Gametogenesis, the primary sex organs, the testes in the males and the ovaries in the females produce gametes, that is sperms and ovum, respectively by the process called gametogenesis. Spermatogenesis starts at the age of puberty due to significant increase in the secretion of gonadotropin releasing hormone. The increased levels of gonadotropin releasing hormone then acts at the anterior pituitary gland and stimulates secretion of two gonadotropins, luteinizing hormone and follicle stimulating hormone. Luteinizing hormone acts at the Leydig cells and stimulates synthesis and secretion androgens. Androgens in turn stimulate the process of spermatogenesis. Follicle stimulating hormone acts on the Sertoli cells and stimulates secretion of some factors which help in the process of spermiogenesis. Spermatogenesis in testes, the immature male germ cells spermatogonia produce sperms by spermatogenesis that begins at puberty. The spermatogonia present on the inside wall of seminiferous tubules multiply by mitotic division and increase in numbers. Each spermatogonium is diploid and contains 46 chromosomes. Some of the spermatogonia, called primary spermatocytes, periodically undergo meiosis. A primary spermatocyte completes the first meiotic division, leading to formation of two equal haploid cells called secondary spermatocytes, which have only 23 chromosomes each. The secondary spermatocytes undergo the second meiotic division to produce four equal haploid spermatids. Spermatids are transformed into sperms by the process called spermiogenesis. What is spermiation? After spermiogenesis, sperm head become embedded in the Sertoli cells and are finally released from the seminiferous tubules by the process called spermiation. Sperms released from the seminiferous tubules are transported by the accessory ducts. Secretion of epididymis, vas deferens, seminal vesicle and prostate are essential for maturation and motility of sperms. The seminal plasma along with the sperms constitute the semen. The functions of male sex accessory ducts and glands are maintained by the testicular hormones androgens. It is a microscopic structure composed of head, neck, a middle piece and a tail. A plasma membrane envelopes the whole body of sperm. The sperm head contains an elongated haploid nucleus, the anterior portion of which is covered by a cap-like structure, acrosome. The acrosome is filled with enzymes that help fertilization of the ovum. The middle piece possesses numerous mitochondria, which produce energy for the movement of tail that facilitates sperm motility essential for fertilization. The human male ejaculates about 200 to 300 million sperms during a coitus, of which for normal fertility at least 60% sperms must have normal shape and size and at least 40% of them must show vigorous motility. Oogenesis, the process of formation of mature female gamete is called oogenesis, which is markedly different from spermatogenesis. Oogenesis is initiated during the embryonic development stage when a couple of million gamete mother cells, oogonia, are formed within each fetal ovary. No more oogonia are formed and added after birth. These cells start division and enter into prophase 1 of meiotic division and get temporarily arrested at that stage called primary oocytes. Each primary oocyte then gets surrounded by a layer of granulosa cells and is called the primary follicle. A large number of these follicles degenerate during the phase from birth to the puberty. Therefore, at puberty, only 60,000 to 80,000 primary follicles are left in each ovary. The primary follicles get surrounded by a more layer of granulosa cells and the secondary follicle soon transforms into tertiary follicle, which is characterized by a fluid-filled cavity called antrum. The theca layer is organized into an inner theca interna and an outer theca externa. It is important to draw your attention that it is at this stage the primary oocyte within the tertiary follicle grows in size and completes its first meiotic division. It is an unequal division resulting in the formation of large haploid secondary oocyte and a tiny first polar body. The secondary oocyte retains bulk of the nutrient-rich cytoplasm of the primary oocyte. The tertiary follicle further changes into a mature follicle or Graafian follicle. The secondary oocyte forms a new membrane called zona pellucida surrounding it. The Graafian follicle now ruptures to release the secondary oocyte or ovum from the ovary by the process called ovulation.